because that's actually very sad. Okay, it is my pleasure to introduce the fellow behind me, who is Professor John Sorkin, and he comes from the Department of Medicine and Department of Epidemiology and Public Health at the University of Medicine at Maryland School of Medicine. I did my postdoc very close to there, so near and dear to my heart. And really is an expert in dealing with biostatistics and informatics. And so this is really an enormous field, certainly with big data and how critical and relevant it is for health and disease. So please, thank you for coming.
So I would like to thank Mira for inviting me. I promise I'll be very short because I told Mira I would be very short. I don't want to keep you late. I've got a timer. When it goes off, I'll try to stop. All right, so first a few words of thanks. Im tirtzu enzo agada, the im tirtzu agada, hi agada tiasher, which means in English, if you will it is not a dream, and if you do not will it, it will remain a dream. We owe thanks to Mira, who willed this conference into existence. Mira, thank you. All right, so I'm going to ask a question. Can genes that affect survival under extreme conditions inform efforts to extend the human lifespan? This is part of a much longer talk that I give, but in the interest of time, I've gotten rid of 95% of it. All right, the longest documented human lifespan is 122 years. The fortunate survivor was Jean Clement. She died in 1977. There is a suggestion of a link to longevity in her family. Her father survived to 93, her mother to 86. Her brother survived to 97. 24% of her immediate ancestors survived to age 80 versus 2% of controls. There's also some investment advice in her family. In 1977, André Francois Raffet, a middle-aged lawyer, offered Madame Clement 2,500 francs per month, approximately $500 for the rest of this 90-year-old woman's life, in exchange for ownership of Madame Clement's apartment at her death. Raffet died 30 years later before receiving title to the apartment, and his poor wife had to continue making the payments. When Madame Clement died at 122, she had been paid about $180,000, twice the apartment's cost. All right, how can we extend lifespan? There is one intervention that has been reliably shown to extend lifespan, and that's caloric restriction, which I claim is a form of extreme conditions. Caloric restriction is a low-calorie diet. It's not a deficient diet. It's supplemented as needed. Generally, you get somewhere between 20 to 60% fewer calories than ad libitum intake, and it has been shown to increase lifespan in rodents by 50%. Pretty remarkable. But is that the only place that it's been seen? The answer is no. It's been shown to extend lifespan in yeast, in rotifers, those are microscopic aquatic animals, and spiders, worms, fish, mice, and rats. So the question is, will this work in higher forms of life, such as primates? Well, there were two studies done in non-human primates. One is in Wisconsin, where extension of lifespan was shown, and another at the National Institute on Aging, where there was no life extension. What does caloric restriction do? It delays the onset of kidney disease, neoplasia, autoimmune diseases, diabetes. It slows down the decline in psychomotor and spatial memory, and it slows down reproduction. It delays it. A big question that's been asked about caloric restriction is, do you have to be on caloric restriction all your life in order for it to work? This was answered in Drosophila, where Drosophila were studied under two conditions. In fact, three conditions. In the red, we see the ad libitum fed Drosophila. On the x-axis, age in days, and the y-axis, the mortality rate. In the black on the bottom of the slide, you see the mortality rate in the Drosophila that have been calorically restricted from birth. And you can see a big difference between the two. Now we take the fruit flies, and at midlife, we subject them to caloric restriction. And you see that their mortality rate comes down to the same level as that seen in those that were restricted from the beginning of the life, showing that you can start caloric restriction significantly after birth and still do very well. What does caloric restriction do? It's been shown that it reduces oxidative damage. That is, it reduces the production of reactive oxidative species, sometimes called ROS. It increases the ability to detoxify reactive oxidative species. And it does that by increasing the production of superoxide dismutase. It also increases the turnover of oxidized proteins, which are bad for you, and it decreases the production of advanced glycation end products, which are also bad for you. Are there any evidence that caloric restriction may work in human beings? And the answer is yes, and it's shown on this figure. We have two panels. On the left, we have data from the National Institute on Aging primate study in male rhesus monkeys. And in the other side, data from the Baltimore longitudinal study of aging, also from the National Institute on Aging. We're looking at three measures: temperature, insulin, and DHEAS. And you can see data for ad libitum fed monkeys versus caloric restricted. And you see that the temperature is lower in the caloric restricted, the insulin is lower, and the DHEAS is higher. Now let's look at data from the Baltimore longitudinal study of aging. On the x-axis, this is survival time in years. The y-axis, cumulative survival. And you can see that among the male members of the Baltimore longitudinal study of aging, those who had lower temperature, similar to this, had longer survival. Those who had lower insulin, similar to this, had longer survival. Those who had higher DHA, similar to this, had longer survival. Therefore, changes that are brought about in these primates by caloric restriction are seen in longer-lived individuals. Might work in humans. All right. We know that caloric restriction improves survival in non-human primates. These are data from the Wisconsin study. Remember, I told you there were two studies: the Wisconsin study that showed increased survival and the National Institute on Aging that did not. Oops. And you can see information on the dietary restricted animals versus the normal, shown in red. This is age, and this is fraction surviving. And you can see that the fraction surviving was higher among the dietary restricted than in the normal fed monkeys. In fact, they did better than hyperinsulinemic monkeys, and they even did better than diabetic monkeys. That's not surprising. All right, so caloric restriction increased survival in non-human primates by reducing the incidence of diabetes, cancer, cardiovascular disease, and brain atrophy. All right, so how might this work genetically? In the Rosetta Stone for longevity studies, at least in terms of genes, is the C. elegans, which is a little worm in dirt. C. elegans and genes that control longevity. Here on the left we see a young worm, and on the right we see an old worm, and if you look carefully, the old worm has got all sorts of wrinkles. Maybe it would do well to use some Ahava products. It turns out that you can extend the lifespan of a C. elegans worm by turning off the gene that is called DAF2. On the x-axis I have age, and the y-axis fraction alive. The red shows the fraction alive amongst the worms where DAF2 is turned off versus the wild worms in which DAF2 is turned on. Is this at all important for you and me? The answer is yes. Why is it important? Because the DAF2, the receptor that is encoded by DAF2 has homologs to human receptors. It has a 35% amino acid identity to the human insulin receptor, the human insulin-like growth factor receptor, and the insulin-related receptor, suggesting that this finding in worms might have analogs in humans. But it turns out that the story is much more complicated than just saying DAF2. It turns out that you need not only DAF2, but you've got to do something about DAF16, another gene. Here is the worm I showed you before, in which DAF2 was turned off by human beings, and the DAF2 was on. DAF2 is usually on in the worm, and here's their survival curve. Here is the wild worm, where DAF2 is typically on rather than off, and DAF16 is typically on. And you can see that we can compare that with worms where DAF2 is on and DAF16 is off, DAF2 is off, and DAF16 is off. The only time the worm does better is when DAF2 is off and DAF16 is on. So you need both of these genes to work together. DAF16 is a 4-CAD family transcription factor. How does it work in the wild? In the wild, DAF2 turns off DAF16. By turning off DAF16, it stops a phosphorylation cascade, and that cascade normally prevents DAF16 from producing a downstream signal, and it's that signal that results in longevity. Turning off DAF2 allows DAF16 to produce its signal, resulting in longevity. It's interesting to look at the life cycle of the worm. Here we see an egg becoming an L1, L2, L3, and L4. These are immature forms of the worm, becoming the adult worm that then makes eggs and starts at the beginning. All right, I will stop very shortly. This is what happens in a good environment. In a bad environment, DAF2 is turned off. The worm enters the dower stage. And when the environment gets better, DAF2 is turned on and the worm continues to age normally. How does this work? There seem to be environmental signals that enter the alimentary tract of the worm. When the worm finds lots of food or doesn't find food, it will produce an insulin-like hormone, which will bind to the DAF2 receptor, turns off DAF2, and that allows DAF16 to work. In a good environment, DAF2 turns off the production of DAF16, which does not allow longevity to occur. In a bad environment, signals that come through the alimentary tract turn off DAF2. That allows DAF16 to produce its signal, which results in longevity. So DAF2 extends lifespan. It turns out it's more complicated than just DAF2 and DAF16. There are a lot of other genes, as shown here and in this cartoon. Is it relevant to humans? DAF2 encodes for a receptor that has homologous to the human insulin receptor, insulin-like growth receptor, and the insulin receptor-related receptor. Some of the other genes that are included in the pathway are AGE1 and DAF2, which also have human homologs. So let's get to the end. I started off by asking the question, can genes that affect survival under extreme conditions inform efforts to extend human lifespan? And the answer is yes. Alterations to genes or cellular pathways that modify metabolic rate and substrate utilization may be the key to interventions that increase longevity. That's it. Everybody go have some food and have fun. I did it.
That means that you have to stay here in this area if you want to be, old enough. Longevity.
Are there any questions, despite this? No. Yes, please.
No, you ask in lunch.
Okay. Yeah.
I'm short,
Okay.
so I'll ask short questions out there.
Okay. Well, thank you very much for that session, everybody, and enjoy your lunch. Betavon.